Hi, Dr. Chrissy. It's good to see you again. Today, I want to talk about the levels of chromatin packing. So we will cover 5 levels of chromatin packing from the smallest level to the highest level. First of all, I'll talk about the first level, nucleosomes. The primary protein components of chromatin are histones. Histones in chromatin structure has high content of amino acids, lysine and arginine, which give them a strong positive charge. So this is histones, and this has a strong positive charge, and the DNA double helix has a strong negative charge. So that's why DNA and histone bind to each other, and they form ionic bonds. The function of histone is to package this DNA into structure units called nucleosomes. This thing is called nucleosomes. Can you see it here? So DNA anchors around the histone and forms a double helix winding around the histone. This wrapping of DNA with histone proteins form nucleosomes, and it's a histone octamer that forms the nucleosome core, and it is constituted with eight histone molecules and 146 base pairs of DNA. This is a so-called beads on a string structure because the nucleosomes are like beads that are attached to linker DNA that looks like a string. Okay, so that was the first level, and this is the basic unit of chromatin structure. And the width of DNA double helix is about 2 nanometers, and the width of nucleosome is about 10 nanometers. The second level is called 13 nanometer chromatin fiber. Multiple histones wrap into a 30 nanometer fiber consisting of highly packed collection of nucleosomes. So there are a lot of nucleosomes here. The width of 30 nanometer fiber is about 30 nanometer. This is about 30 nanometer. And current models suggest that nucleosomes of the 30 nanometer fiber seem to be packed together, and it forms a regular and three-dimensional zigzag structure so that it can fold together with its neighboring fibers. And the next level, the third level, is called DNA loops. The 30 nanometer chromatin forms a string, and this string becomes many loops. You see those many loops. They're placed next to each other. And the width of the loops formed by highly compact chromatin is about 300 nanometer, which is the length of about 50,000 to 100,000 base pairs. Base pair is a pair of complementary bases in a double-stranded nucleic acid molecule consisting of a purine in one strand linked by hydrogen bonds to a pyrimidine in the other. And then the next level, the fourth level, is called heterochromatin. Heterochromatin is composed of many loops, the DNA loops, of 30 nanometer chromatin fiber. There's another chromatin, which is loosely packed, diffuse chromatin, and that's called euchromatin. And the width of this chromatin is about 700 nanometer. And the last level, the fifth level, is highly condensed, duplicated chromosome of dividing cell. Highly compacted arrays of heterochromatin eventually form the centrosome, and that attaches the two chromatids together. So this is a chromatid, and this is a chromatid, and that's a centrosome. Actually, centromere that connects the two chromatids together. And the width of the duplicated chromosome of dividing cell is about 1,400 nanometer. So this is basically how this chromosome is formed. It starts out with a very small structure called nucleosomes, but then this nucleosomes form chromatin fiber here, and then that fiber forms a loop, and then that loop eventually forms heterochromatin, and then this heterochromatin forms this nucleosome, and we have 26 pair of this nucleosomes in our human genome, and since it is formed by the DNA double helix, our DNA is kept very securely, and also it expresses the gene that's stored in our DNA in an efficient way. So this is how we can see that this is a way we can see that DNA actually is a structure, is a part that forms the structure of the chromosome. Thank you very much.